So what I'm going to do today is talk about more from a very higher resolution molecular interaction point of view, rather than larger, larger uh, physical based discussion that has been taking place. And I will try to show why very fine scale molecular interactions, how they may be determining a lot of things that we see at the much larger length and time scales. Um, and the title of my talk is Low Complexity Domain Reinforces Specificity of RNA Binding Domain. Okay. Before I start, let me thank my lab. Almost all of the work that I'm going to show you has been done by Sangeeta, who is not in the picture, and also by Krishna, who just graduated. And I have to thank all my funding agencies for providing this support. Okay, so I'll straight away jump to it. This is the only slide that is sort of introductory slide, and this I'm using to pose a few questions to all of you. Okay, and so this slide is taken from my collaborator Sobhamai Maharana, who is currently in Biophysical Society meeting in US, and this is a very this all of you are familiar with the left hand side picture where you see all these membraneless organelles both in the nucleus and in the cytoplasm, and because the biologists have been able to identify them as separate, there has to be some distinguishing features between these droplets, and that is why they are named differently, like you have P body, U body, stress granules, and so on and so forth, right? Some of them have been known for ages. For example, Kahal body and others. People have been knowing about their existence for ages. For example, uh, one of the very well known marker for dementia and Parkinson disease was a Lewy body, which has been known for a while that it was connected to Parkinson, but it was only in the about 20 years, 25 years back that people connected that to alpha-synuclein, and that Lewy body was actually made up of alpha-synuclein and rich in that, and that led to that was the time when it became clear that these are rich in IDPs and proteins that are of low complexity domain, which I'll talk to you more about. So that is one point. The other point is that I know the title of the meeting that Devayan and Sandeep has chosen is called liquid-liquid phase separation. But I believe the jury is still out on whether it is a purely density-density phase separation, or, and there have been recent discussions on higher order kind of organization and and theories like percolation theories, PCPs, you have theories where it
Can
is.
you hear us? Can you hear me?
Yes, we can.
Yes. So actually, the we don't have liquid phase separation in the title. It's really biomolecular condensate. We wanted
Excellent. I
to
like that.
make that clear because I think
That's very good
so
because
that confusion is not there.
you can see that there's viscosity in this, like there's a shear flow, and these are definitely there are networks getting formed, and they eventually, based on interactions at the molecular level and other factors, they can actually also become very jellish and solid likes, and so on and so forth, and they are also separated. So it's good. I'm very happy about this. The third image that I show, a movie file, is about the organization. So these there is I don't remember exactly which protein. I think it's the first protein that Soba has tagged here. You can see there is a heterogeneity in the distribution of from a very high concentration to very low. So there is a overall heterogeneous organizations of proteins and RNA in these systems also. So the big questions in the field, I would say, are if you have to, like, which is not very easy and probably very formidable and impossible to answer with just theory and modeling, as we all know. But together with experiments and theory, probably there are many things that could be addressed. So the question is, if there are so many similar condensates, what makes them specific? For example, how is P body different than stress granule? Why are they localized in different regions of the cell? If it is just a physical based principles of condensate formation, then what localizes them in certain areas or certain regions of the cell? How are their compositions different? Because they have to be functionally connected with it. They are, and how is that composition determined for them? Like, are they rich in certain proteins and RNA? Do they have a certain specificity for certain RNA that they are trying to protect from degradation, or like, and also the physical properties. Like, if you are able to, let's say, probe it, and look at things like viscosity or their viscoelastic properties, would they all show similar behavior, or you would say you would see differences in them? Would you see differences in them at different point of time as the cell matures or in the cell cycle? And so these are the big questions in the field, particularly from the point of view of biological functions and the intracellular organizations of them. Of course, I cannot answer all of them, but we are trying to kind of delve into some aspects of that. Okay, so just for the students in the audience also, these would be the questions that would really be valued if you are able to attempt to answering all of this. And so I am going to discuss more about the molecular rules of phase separations for specifically for first protein, because that's the protein that we most closely work with Soba's lab. And this is also a slide that probably many of you have seen in the past. So these phase separation, these condensates that I show here, are actually composed of RNA binding domain. So most of the proteins that we you work with in these systems are proteins that have a multi-domain characteristics with a RNA binding domain, or what is also called as RNA recognition motif, and there is a prion-like domain, or you can also call them as low complexity. There are subtle differences between the two, but the low complexity domains are the domains that have sequences that have frequent repeats and have and they're low complexity in the nature that the number of permutations and combinations are limited as compared to usual folded protein systems. And they are the condensation is driven by a lot of factors, and also very weak interactions like cation pi become very important in all of these systems. So this is a schematics that kind of tries to show how you have on the left hand side. So this is low phase separation tendency or low condensate formation tendencies to a high condensate formation tendencies, and you can see the kind of interactions that are driving this. So you have pi interactions, which is a weak interaction but important interactions. You have a cation pi interaction from phenyl. The pi is coming, lysine is providing the cation, and then the level of cation pi's could be different. You have, for example, a tyrosine lysine will be a stronger cation pi interaction, and you could have a multiple of them combined together. So in short, what I'm trying to say is that what we see at very large scale micro or sub micro meter scales in cells are driven by these very fine interactions at the molecular level. And so I'm going to answer one particular question in this, and that is like, what is the first RNA interaction? How does and this is in particular with respect to stress granules. So this is a movie of a coarse grained molecular simulation of first molecule. And let me first explain to you, because we will be talking a lot about first molecule. You have a RRM domain, which is the RNA recognition motif domain, classically known to recognize certain RNA that the stress granule or the first would sequester it into the stress granule, for example, or in any other compartment. And on flanking on, so the pink one is the RRM domain here. Okay, and so this recently we did some work where we have actually taken the Sachs profiles and any other experimental information that we have and developed this model um, to kind of show the conformational landscape that we are capturing correctly. And with Devayan, we are kind of pushing this further to bring in more subtle interactions into this with Santosh and Devayan, and also have RNA models that can interact with. But here I'm showing this movie just to give you a structural feature domain architecture of RNA of first. So the pink one is the RRM domain, and the zinc finger is the blue one that you see. And in between all of them are these different stretches of low complexity domains that are disordered proteins. So you have the SYGQ, what is classically called as the LCD domain of the FUS, which basically is the one that mediates phase separation. Then you have on the N-terminal side, you have the RGGs1, which we will talk about soon. And on the other flanking side of the pink module, you have the RGG2, zinc finger, RGG3, and there is towards the end there's a nuclear localization signaling sequence that actually makes sure that the FUS is most of the time localized in the nucleus. Okay, so briefly, this is what FUS structure and structure as I'm showing you here, and the modular sequence modularity looks like. Okay, and this is the data that we started working with. Okay, and this is very fascinating data to us. So if you take the first molecule and bind it with RNA, you get a binding affinity of. So this is the data from Jacob Swartz lab from Arizona University, who did a lot of work when he was with Tom Check on RNA protein interaction, and this data is particularly with FUS RNA interaction and a particular type of FUS that is used to express HNRNP1 protein, which we are not going to discuss at all. Just know that FUS likes to bind to a certain RNA that will be used to express certain protein in the cytoplasm. And so, in other words, FUS is trying to sequester a certain RNA with itself when it is forming biomolecular condensate. And so you have wild type FUS, which has a binding affinity of zero point 0.7, quite micromolar though, and the RRM part, which is thought to be the RNA recognition motif, has a poor, as compared to the full FUS, has a poor binding, which is 90 micromolar. And they showed that if you have RRM and on both sides are RGG1 and RGG2, so if you just take the RRM and flank it with these two RGG1 and RGG2, the binding affinity jumps from 90 micromolar to 1.9 micromolar. And to me, this is significant because it basically is telling you that the folded domain is giving you a 90 micromolar, and the disordered region, which is not thought to be very strong binders, are jumping the binding affinity by about 100 times better, or at least 50 times better here, right? And so and so these are all ITC data. So we wanted to understand what is the molecular origin of all of this. And you can think of it in terms of design principles. You can think like, suppose you wanted to make an in vitro condensate and you had a bunch of different RNA and you wanted to design a sequence that could only sequester a certain RNA with itself in the condensate. How would you go about? In if for me, this is like trying to understand the fundamental design principles behind the partitioning that may take place at synthetic level, so to speak. Okay. And to do this, what we did is we kind of did a very large scale biomolecular all atom simulations of the first protein with keep adding the with the RNA, and then you take the first RNM domain and add the flanking disordered regions into them, and then try to understand what is the interactions that are coming up and how can we understand whether we are able to explain these large increase in binding affinity. Okay, and so the first thing that we found out in this work, so this work was published last year in Journal of Biological Chemistry. The first thing we found out was that the RRM core, so you can think of this as the core of the RRM molecule, and they actually do not have very strong binding, as we know, 90 micromolar. So right away you can see they start dissociating, and this profile. So the way to read about this would be, this is the minimum distance between the RNA and the protein, and this shows, so the y-axis on the bottom graph, the dark orange shows the two stem of the RNA, and the middle one shows the loop of the RNA. Okay, so it's a stem loop construct, and wherever you see more brightness, those are the interacting side. The bottom is decorated with the structural motif of the first molecule, whatever we are working with. What we found out was that the moment you have a small c, the boundary residue, which is the start of the basically you have a region around here, around 377. Those we have identified as boundary residue. The moment you have this boundary residue, you can see this as a small, transient helix here. The binding actually becomes quite stable, and you can further. So we showed that those are the most important motifs for the protein to be bound to the RNA. And so this was very interesting because generally when you think of RRM and the LCD domains, you do not think about what is happening at the boundary. Could that be so important that if you mutate the boundary residues, the binding does not take place as strongly as it would take place with the folded part of the protein? Because generally the protein RNA interactions, you think of them getting stabilized by the folded part rather than the disordered part. So this was one finding which was quite interesting, and you can see that we took this and did further studies on this. So I'll talk about that soon. Okay.
Hear me? Anand, can you hear us?
Yes, yes.
Yeah, I have a question.
Yeah, please go ahead.
So, is it possible that the RRM domain, the RNA recognition motif, is providing specificity, but additional affinity without specificity is being provided by the low complexity domains?
Both. So you are right. So actually, it's both ways. We showed. So I'll show you some data that actually. There is a very low affinity recognition, and further stabilization is coming from the RNA. But then what we did is once it recognizes. So we have done. Let me come back to your question shortly because in interest of time. But that is exactly what we found out and answered. Yeah, yeah. That's right. So so again, so let me come back to the, you know, sorry, I may not be able to get your name, so please excuse me for that. Can I know the person who was asking the question?
The extension of your name, you are Anand, I am Purnanand.
So you are complete Anand, complete bliss. I am just blissful, so sad for me. Punanand, so great question, Punanand. I'll come back to it.
Yeah,
And so,
one more thing. So I'm also chairing the session. So you're 19 minutes into a 30-minute talk.
okay, I will be quick. Yeah, yeah. So I'll quickly go through all of this. So then what we started looking at is what, uh, you know, how much does the repeat of the RGG so you would see like here, right? When they had a construct where the RGG2 part, the second, the right, the C terminal part of the protein, you can see like when they add a stretch of RGG or disordered region, you have more or less similar, experimentalists would call this similar micromolar affinity. But at some point of time, there's a flipping that takes place. And so this was also quite interesting. Like this goes at the heart of like what would be the optimum length of the disordered region that you would have between, let's say, two folded regions, or like when do they start actually kicking it in? And so here we found out that when you have the larger 390, which is all the three RGGs boxes of the RGG2, then you start seeing more interactions with the RNA. And so what is happening is that there is a region that is not getting because of the structural stericity is not getting interfacing with the RNA. And that, so I can answer those questions later in the detail. So in interest of time, I'm rushing through. And then we looked at closely at the residues that are there and sort of kind of rationalize this behavior in terms of interactions of the RGG to the further stretches of RGG are interacting better with the RNA. Okay, and because we had developed an algorithm that can cluster IDPs, which I'm not talking about today, we actually clustered the whole interaction, and we showed that there was a part of the, so you can see like there is a part that is interacting and then the part that is away from the membrane. So we could quantify all of them and rationalize how why the longer stretches were interacting better with the RNA or not. Okay, and then this is the another interesting part we saw. So when you put both the RGG1 and RGG2 on flanking side of the RRM, you see this very interesting feature. You plot the interaction, you subdivide the interactions of RRM with the RNA, RGG2 with the RNA, and RGG1 with the RNA. And you could immediately see that the RGG1 doesn't even bother about interacting with the, just the stem part of the RNA. It is, sorry, the loop part of the RNA. It is basically more focused on the, because it cannot, it doesn't have the space to interact with the RNA at the stem region. It actually has interaction with the RNA. So now, and we also showed that the RNA was more stable when you have the RGG1 in the system. So that essentially shows that the longer disordered region was stabilizing the RNA, and the RNA protein interactions was further stabilized by these very weak interactions but persistent interactions with the disordered region. And you can see how it forks out. Like you can see the RGG1 is on the far side of the intensity is on the far side. So they are basically clamping on the ends of the RNA, and this is on the near side of the stem loop region, the RGG2. So clearly there is a division, and that you can go back and look at the sequences and the sequence grammar of the LCD, and probably get some more information out of that. Okay. So I guess I will push through. And then we started thinking like, how do you define the, because that's the story that we are trying to say. We are trying to tell that please do take into account the regions that are considered to be the boundary residues. And then Sangeeta went through and looked at a vast set of literature. So first RNA interactions have been in play much before, much before LLPS and biomolecular condensates became, fashionable, and people started working on it. And we could see like, so just focus on the regions, how people are defining the region between RGG and RRM. And you could clearly see that there is a huge amount of ambiguity in how would you define what makes the RRM, and where does the disorder LCD part start. So you can, and so that again boils down to the point that there is something interesting happening, and you clearly don't know like what they could be doing. And our work in some ways tries to make the point that the boundary regions are important for binding. Okay.
I'll interrupt you for a minute here.
Yes.
Pulshat has raised his hand. He wants to ask a question.
Yes, please go ahead.
But
Hi,
since you have 5 minutes in which to wrap up and also answer questions, I'll request Pulshat to put it off till you wrap up.
Yeah. I'll wait till he finishes. No problem.
So
Yeah.
please try and finish in about.
I will. I have a couple of slides more. So then we looked at the moment you put, so if you look at the cavity of the RRM for RNA, let's say, the moment you put the C terminal region, the small stretch, you suddenly, it's like it opens up and provides like a large pocket. If you don't have that, then it doesn't have that kind of pocket. And this is the interesting part, that the transient helical motif in this domain, in the boundary region, actually provides. It provides a larger pocket for the RNA to sort of engage with the RRM. And so this is, and then we looked at all the kind of interactions that were happening. And very, very interestingly, you find that there are residues that are, for example, look at this. So the boundary region has a highly multivalent interaction. For example, the arginine 372, this is the true multivalence. You can see it is forming cation pi interaction. So it can form multiple forked cation pi or pi interactions. Sorry, not pi cation or salt bridges interactions with different residues. And so we could clearly see that this was an important hub for the RNA protein interactions. If you look at the nucleotide RNA interactions in this. Okay. And then, so for experimental purposes, we then went and mutated these TRR. So there's a small stretch called TRR there. So we mutated it to alanine. So we did, because the experiment was easy to do with triple mutant, we mutated individually and triply also. And you can immediately see the RNM. And this goes back to your question, Purnanand. The RNM RNA interaction is this is the wild type. you mutate the TRR, which is actually at the boundary residue. They are not the canonical binding site. You clearly see the pink one. I'm sorry. You see a shift in the RNA protein interaction. So the RNA protein interaction is getting weakened if you actually, so if you are able to bring the protein to the RNA, but you don't have a strong TRR interaction, it is very loosely bound to the RNA. There is a shift in the RNA protein overall distance. And similarly, we have done, looking at how that affects the RGG and RGG1 and RGG2 interactions. That doesn't seem to matter so much. So the disordered regions keep interacting the same way. The RRM actually weakens its interactions if you make the TRRS triple mutant. Okay, and that is something that Sobha lab have been doing in their cell biological construct, where we see that the recovery time weakens when you have the first TRR interaction, which essentially tells me that the network has the recovery time actually gets better, which means that the network is kind of now more loose. And she's doing, see, they are trying to actually, because these are cell constructs and they are much difficult to interpret because there are so many other activities happening. So they are trying to do a in vitro reconstruction of first RNA with a wild type and our mutations, and trying to so I will summarize this and take up the questions later. So residue at the boundary of ordered RRM in disordered region is another feature in the RRM that we should be thinking about, and thinking like how does that further improves the binding affinity. So it is like accentuating or consolidating the specificity to a stronger level. And and as a multivalent interaction, I did not show the data, but we actually mutated or replaced one nucleotide of the stem region with another, and we show that the binding is lost. So there is a certain specificity to the RNA sequence and shape too. And this is what is our bigger question that we are asking with SOBA. Mutation at the boundary residue should affect the stability of RNA hairpin, while RGGs continue to interact with the dsRNA. So you may not be able to sequester the RNAs that you want if you mutate these regions, and you may actually become more promiscuous in binding to different kinds of RNA. And so just I thought I should also put a future slide. So right now with Neha, who is also in the audience, and Santosh, we are trying to sort of our plan in the long term, short term, whatever you want to call it, 3 to 5 years, is to actually be able to in silico reconstitute the stress granule by doing a lot of getting data from proteomics and any kind of experimental data, so that you have some sort of in silico system where you have RNA and proteins, and then you can probe for how the properties, viscoelasticity and other properties change when you change the compositions and so on and so forth. And so for that, we are building up coarse-grained models of RNA, protein-protein interaction, protein-RNA interaction. These are some of the coarse-grained models, and currently working also with Devayan to develop a RNA model that we can couple with this and kind of study the different levels of physiological and pathological constructs in the stress granules, particularly, and P-body, because with Raj also we are been talking for a while, and that has been quite a good collaboration there too. So with this, I will end and take up questions.
Yeah, thank you, Anand. We'll just take one question from Punjab and move on to the next talk, if you don't mind.
Yeah, Poonanan, can I ask the question?
Yeah, go ahead. Very quick.
Okay. Hi, Anand. Wonderful talk.
Yes,
I was actually hoping that I'll get to meet you here because, we have not managed to meet in a while on campus. So I was looking
yes, we should be meeting soon.
Yes.
Yep.
So, all right. So I have a comment and a question. A quick comment that in Jacob Schwartz's data, the outlier is actually the RRM only construct, right?
Yes.
The rest of all the other constructs are kind of comparable in terms of their binding affinities, right?
So let me pull that out. So if you look at you have the RRM, and then if you add first to RGG, then you have like still a little bit similar, you can say similar. And the moment you have the RGG longer, then you start seeing the jump.
Right. So my point here was I forgot what RNA was used in this experiment.
So this RNA, so this is the stem loop RNA, and those are the RNAs, those are the mRNA for the HNRNP1, some part of the HNRP1. I have the sequence somewhere. It's in the page.
Sure, it doesn't matter. My comment was that, to begin with, we don't know if this is a good substrate for RRM, right? Because despite their broad specificity to bind RNAs, there are some RNAs that RRM binds better than the others. So I'm wondering if one took a substrate which is good, which has a good binding affinity for RRM, would these numbers alter?
Yeah, you are right. So yeah, so the in terms of the RNA strength of the RNA binding, this one is, I would say, this RRM would be a poorer recognition motive in terms of the, because it's a micromolar affinity, right?
Yeah, yeah.
And
But too high.
it's too, the values are too high, which is weak binding, right? So we found out that these are really not a very strong binder that what you would see with some other RNA, and that would be an exercise to do, like take a RRM that has, let's say, some sort of a nanomolar or a tenths of nanomolar binding, and see if at that level the RGGs and the LCDs would further accentuate it or not, right? Or they don't matter.
Yeah.
I think this is where it is becomes interesting, because if they are very strong binder, then the whole thing becomes too specific anyways, right? And so the room for you probably don't need the LCDs to further consolidate the binding there.